So as I mentioned in the just opening there, this morning's panel was to engage, and very fortunate to have really a range of therapeutic areas represented here, and a range of successful organizations going through the various stages of clinical trials, to engage with you all in a little bit in how we might think about engaging with stakeholders, what may happen as we go through these processes. As we observe somewhat in pharma, while pharma success rates have certainly been coming up, and we may characterize that to increase in some of the more understanding of mechanisms as we move into more protein drugs, we still see a little in cell therapy, a difference compared to regular pharma, where in cell therapy, if we see a failure, sometimes it seems like it's the end of the company, as opposed to big pharma with small molecules, it's just that's part of the business model. So how do we anticipate a little bit as we go through this? What does it mean, and how do we interact? So that'll be engaging in the panel. What I'd like to start to do is just start by an introduction to the panel members and ask each panel member maybe to run along the row and introduce themselves to you and say a little bit about what you're working on so you can get a little bit of context to who we have on the panel. We'll start with you.
Thank you. I'm Donna Scarrett. I'm Chief Medical Officer of Mesoblast. Mesoblast is a regenerative medicine company that was founded 10 years ago. Our technologies are predominantly cell-based, and we are now in late-stage development in a number of indications related to chronic degenerative diseases.
I'm Gil Van Bockelen. I'm Chairman and CEO of Athersys. And Athersys is also very heavily focused in the area of regenerative medicine. We've been focused in that area now for coming up on 14 years. And have 5 active clinical programs across a range of different disease areas, which I'll be talking about a little bit later today in my presentation just before lunch.
Great. I'm Gwen Binder-Scholl. I'm the Executive Vice President of Adaptimmune LLC, which is the U.S. arm of Adaptimmune located in Oxford. We focus on T-cell immunotherapy and oncology, and we develop affinity-enhanced T-cell receptors. We currently have one product against MISO antigen in the clinic in multiple oncology indications. And we have multiple other TCRs coming through to the clinic about mid-next year.
Good morning, I'm Jay Siegel. I'm Chief Biotechnology Officer and Head of Scientific Strategy and Policy for Johnson & Johnson. Johnson & Johnson is very broadly based in healthcare. I personally have been engaged in biological drug discovery, development, and regulation for the entirety of my career. J&J is actively engaged in regenerative medicine. Our lead product in that area is a cell therapy for retinal disease.
Good morning, my name is Christian Homsi. I'm the CEO of Cardio3 Biosciences. We're working mostly in the heart failure space, and also to a lesser extent in the acute myocardial infarction space. We work with cells on the heart failure space, work with cells that are reprogrammed to become cardioreparative cells. And this is technology that we acquired from Mayo Clinic about 7 years ago, and the company was founded 8 years ago.
Thank you, everyone. So, well represented across the groups. Maybe I'll start with Gil. As we were thinking about this panel, you prompted me to a couple of articles that we looked at in Biomed Tracker that said really the increases going on here in new approvals. We had 97 new drug approvals in the United States in 2013. We get the most approvals going on 22, 23. The industry's looking positive in that way. Nature Biotech had an article earlier in the year, and also highlighted the regulatory success rate for biologics is nearly twice as high. And so somewhat, maybe I start on a positive theme. Do we pick up, Gil, from that article that biologics, while there's a lot of risk inherently, because they are biologics and we know more about them, is it a better place to start than we're starting with a small molecule? Is that a part of the argument we use here when we think about biotherapy? Can I start with you, Gil?
Yeah, I think that's right. The numbers over the past decade, I think the group at Biomed Tracker and the Sagent group that have done a tremendous amount of analysis in this area and have been tracking things, actually thousands of different drug development programs and biologics development programs from around the world over more than a decade, illustrate pretty clearly that the success rate in biologics is meaningfully higher than it is for traditional pharmaceuticals. Now, one line of thinking would suggest that because many of these therapies are materials that originate or derive from things that are naturally in the body, that perhaps lends them with an advantage on the safety side. And I think the statistics actually bear that out. I think that there's a couple of key points on the regulatory front. The regulatory trends in the past few years, as you were describing, Mark, appear to have been improving pretty meaningfully as well. We see success rates that are actually going up. I think that that's a good indicator, and it really illustrates that the regulatory mindset has fundamentally changed and that there's been a tremendous amount of information and knowledge gained over the past few years, not just by the FDA, but by other groups around the world that are in the midst of some pretty transformative initiatives. The PMDA in Japan, for example, the EMA in Europe, have all embraced a somewhat different mindset over the past couple of years, which is really looking at how do we make it possible for safe and effective drugs to be developed faster and more efficiently. And I believe that that's actually beginning to translate into some of the statistics that we care most about. One of the things that I think we as a community could do a little bit better is that the cell therapy field in generally, which has many different forms and many different flavors, if you will, has a pretty remarkable track record in terms of its overall safety, which I think is meaningfully better than what we've seen in other types of modalities in the history of drug development. What we have yet to fully establish is the breadth of clinical utility and proof of concept from well-designed, properly controlled trials. But I think it would be a mistake, and some people look at the field and say, well, it has yet to actually prove that it can make a difference in patients' lives. Well, we have to look no further than the statistic that Jeff actually mentioned this morning, where Organogenesis and their umbrella of technologies have now successfully treated over 1 million patients with cell therapy. And there are other examples of cell therapy products that are already out in the market, and many more that are coming that I think are really going to transform medicine as we know it. So I think the regulatory landscape improving and some of the initiatives that are now being implemented as we speak really create an exciting opportunity for us, but it would be a mistake for us to think that everything is going to be safe or everything is going to work. And I know we're going to get into that a little bit more in the discussion of the panel.
We will. We'll warm it up with a positive outlook to start the Tuesday morning, and then let's get down. So, it seems like that's positive enough, Jay, to attract Big Pharma into this space. A couple of years ago we had the panel with Big Pharma present, and now, we see, I guess, a couple of papers come out on immunotherapy and cancer, and some of my panel members signing up here, as well as J&J, well represented. So has pharma been risk averse, or sees this as this is now enough to be in the space?
Well, let me thank you for the question. Let me start first by extending a little bit on Gil's comments. The safety of biologics, including cell therapies, I think comes not just from the fact that they're natural, but they tend to have, compared to small molecules, to be less likely to have non-specific binding to receptors and off-target effects. So we've seen relatively little of that. They do have their own specific toxicities, immunogenicity, the risk of infection. I would add to your list of cell therapies that have been effectively used in millions blood transfusions. I think it'd be pretty silly to reject the notion that cell therapy can be useful when you think about blood banking. The point of your question, there's a great deal of interest in the pharmaceutical industry in immunotherapy of cancer. That spans small, large molecule, and cellular therapies, and I think all are of interest to our company and to all of those companies that are making serious efforts in oncology. I think beyond that, in the broader realm of regenerative medicine, certainly at Johnson & Johnson, we've been engaged since I've been there 12 years in cellular therapy. It's not a big part of what we're doing. It's growing. We've done a number of deals in recent years and partnerships, and I think that there is, quite appropriately, given the data, a growing level of interest. It's going to vary from company to company, but we see it as a significant part of the future of medicine and are currently planning how best to be organized around it and to ensure that we can best take part in opportunities as they arise.
Great. Gwen, maybe sort of following on naturally from that question, as Adaptimmune clearly engaged in one of those hot areas in the immunotherapy portion of it, and recently done something with GSD in a partnership there. Maybe expand on how you're thinking about it. I mean, you've gone from very small company to quickly now scaling up as you look at this. What are you most worried about as you think about next steps as you're on this wave?
Yeah, no, I think that's a great question. I mean, we're in a really nice position now with GSK partnership and also the additional financing that we have to really think about where are our gaps and how can we ensure success in our clinical development. And I think in thinking about how do you define success in clinical development, it's not just do you meet the safety endpoints or the efficacy endpoints, it's how can you design a clinical development program that addresses what you don't know and can add to the knowledge in the field. And so with T-cell immunotherapy, I mean, you really need a high depth and very durable response in our patients in order to bring something forward to commercial to the commercial markets. And in addition, we're dealing with cancer immunotherapy, which has the complexity of the tumor microenvironment, the product variability, and then also the patient's immune system. And so unless we get very lucky in early trials and we hit a home run, we need to really understand those various components. And because of that, really developing a strong translational medicine team and working with companies that have, emerging tools to understand the tumor microenvironment and the immune system so that we can understand what patients will be responding to our therapy and what is that patient population and and also informing how can we improve the therapy as we move forward is something that we need to spend a lot of energy up front to plan and to systematically investigate in our clinical development plan. And so I think that doing that properly will prevent failures in clinical development because we'll learn at each small pilot study along the way. And ultimately, by the time we get to the end of our initial clinical investigations and we're ready to move on to pivotal studies, we'll know the patient population and we'll understand the science a lot better. So I think that's the biggest risk is not understanding the science of these complex systems. And I think, making a strong investment up front into that science is absolutely essential.
Thanks. Turning to you, both Donna and Christian, I mean, as I sort of read, the success of maybe the immunotherapy and where we are at positive, I also worry at the same time, being around long enough when we started this in gene therapy, that a couple of things go wrong and then the whole industry goes down. And in some way, I worry. I mean, you both are in a slightly different area of the cell therapy space. So my first part of the question to you, and you're sort of further down, is do you worry that, certainly if I speak to the local immunologists around the research community here in San Diego, they certainly will tell me we don't know enough about the immune system yet to go down and really do this T-cell therapy. So do you worry that what you're doing in the areas sort of in different stem cells or a different area of regenerative medicine gets affected by maybe a failure in another part, or are you happy with the halo effect of a positive thought on immunotherapy? Maybe start with you, Christian, on that thought.
Oh, thank you for the question. Yes, indeed, it is. The field is communicating with each other, and obviously, any failures or success in one company will also reverberate in the other. Having said that, it is. I would tend to support what Gwen just said in terms of understanding the science at the beginning and understanding what makes a therapy successful or not. Mm-hmm. Some of it is the product, some of it is how you deliver the product, some of it is how the quality of the product when it gets to the patient's bed. It's an equation with many unknowns, and understanding all those different parts. Local drug delivery is very different from systemic drug delivery. The effect on a heart when you inject cells in the heart is not something can be affected by many different things, and so failure is multifaceted. Is it failure, or is it failure of one element of that equation that you need to adjust to get a success? And so that I would say understanding those mechanisms, those interactions between each other, I think that's the critical thing we have to do as an industry. And
turning to you, Donna, I mean, the product is certainly quite advanced, as we've taken stem cells in this original meeting with stem cells in how you think about it. Your own personal history, you've come all the way from transplantation into the stem cells, as we talked about. So expand on how you think about, that question of, are we better enough understanding on the science in some of the area you feel where you are in your clinical trials, and what do you worry most about as you think about what's going on in the rest of the industry at the moment?
Yeah. Well, for one, I think we never fully understand all of the mechanisms and the potential complications of the therapy, but our priority is really to deliver medical care for patients who have needs. And so there's a balance that's needed between our understanding and what we need to try to deliver to the medical community. I think that how we see a failure, I agree, there are many different types of failure. For me, if we learn something for the process, I wouldn't internally process it as a failure, because what we learn helps us to advance the field. And I think that we have an obligation as a community to educate the public to understand what this process is, and that you can and should move forward even if there are stumbling blocks along the way. I guess if you ask me personally, what do I get most concerned about, I think reckless processes are something that I think can be a real deterrent to the field. And obviously, that's not the community that's involved in meetings like this, but there's been a lot of activity that goes on that gets in the news and has potential to set us all back, and it's actually very much unrelated to the type of diligent work that's going on in the development programs from the academic and industry persons involved in this field.
It's a good point. Other the panelists, when you worry about other things going on in the field or these successes, how do you think about what the community we need to be putting out? Certainly we're talking about, here, do we understand the science, and we know there's going to be some grey areas, or lack of understanding, not grey, it's just we don't understand it. But is there a way to design the clinical trial that is the right way to design the clinical trial with the right endpoint? How do you think about that, Gil? You're fairly...
Well, I think it's important to remember that translational science is somewhat imperfect, right? We're taking things that we learned from the best relevant preclinical translational animal models, and then taking that knowledge and then applying it in a human clinical setting. And another complexity is that sometimes the product that we actually want to use clinically may not be the thing that we're actually able to evaluate in the preclinical models, or they have to be manipulated in a way so that it doesn't really perfectly replicate, if you will, the biology that we're intending to explore clinically. I think one of the things that came out of the research that you were mentioning earlier, Mark, and that has actually been alluded to here in various ways from comments by the panelists, is that we learn a lot at phase 2 development. It's no surprise that that's the riskiest stage of clinical development, where we see the greatest amount of attrition, if you will, or maybe attrition is not really the right word, but it's where we see studies that teach us very important lessons, perhaps about we didn't get the dosing strategy right, or the timing of administration when you're having to give multiple doses over time, or the route of administration needs to be further optimized, or there's a whole host of other things that need to be taken into consideration. But not too surprisingly, we see more and more examples of developers, sponsors, clinical investigators that are taking knowledge from those prior studies and then successfully applying it in subsequent studies, because they're learning from the failures, they're learning from the disappointments, they're learning from the stumbles. I think it's important for all of us to recognize that it's only about 1 in 3 of the phase 2 clinical studies that actually hit their pre-specified endpoints, their pre-designed targets, if you will. But that doesn't mean that that the other 2/3 of the studies that are run are failures. It just means that we're learning things that are somewhat unexpected, and a lot of it has to do with the fact that translational process that we're talking about is not perfectly predictable, and so we tend to learn some things about the biology as we go. Cells are especially tricky because cell therapy medicine is multimodal by its very nature, and as Gwen was saying, there are lots of different parameters, lots of different things that we have to take into consideration, and some things that we don't even yet know how to precisely measure, which is why the collection of companies that are developing technologies that support the ability to monitor for potency or biological activity in a variety of different ways are critical to the long-term success of the field. By working together and kind of asking the right questions and then applying that knowledge including from the mistakes, the stumbles, the disappointments, or the things that didn't go exactly according to plan. That's how we ultimately get on the best footing, both from a regulatory perspective, from a clinical perspective, and ultimately from a commercialization perspective.
Well, I would just want to add on to that. I mean, we have a little bit of an advantage because we are looking for that, high frequency of response, and so we have the luxury of building in some of these questions into a series of small 10-patient pilot studies where we can interrogate what is the patient population and what is the delivery route, and basic questions like that before we even move on to a larger phase 2 study.
Kind of POC-light exploratory type studies.
Exactly.
Exactly.
Yeah.
And I'd add to that unlike failing in phase 2, failing in phase 3, you often don't get another chance. It's costly to fail in phase 3, whether you're a small company dealing with venture capital funding or a large pharma dealing with a broad range of development choices. It's very difficult to raise money to study a product that has failed one clinical trial. And as Christian said, the causes of failure are multifactorial, including trial design, as the question indicates, but also the target population, as you were talking about. And also in this class of products, it's often the product itself. Unlike a drug that you more or less have what you have, you can modify the dose with a cell-based therapy, with a regenerative medicine. You also have opportunities potentially to tweak the product. And that's why it's particularly important. There's a tendency often to get to that phase 3 trial, especially when funding is short. But in almost all cases, learning the lesson, understanding the science early so that you can design the phase 3 trial with the optimal chance of success is the surer road to success.
So picking up on that, Christian, can the headline look different to phase 2 trial failed to phase 2 trial we learned something and we designed it in a way to learn it? I mean,
That would be good.
can the audience report that? Is that what the community needs to be able to educate around, that really it is a trial? How do you think about your phase 2 trial?
Well, I think so. I think phase 2 trials are not meant to prove that your product is effective in a broad population. The phase 2 products are meant to guide you into what is your next development stage and what is your most likelihood of success in your next product. So we have to be modest about what we expect from a phase 2 trial. But I agree, we need to spend probably more money than we've had in the past on our phase 2 trials, better understanding how they are. Now, this is an ideal world. Frankly speaking, most of us in relatively small companies do not have access to very large amount of funding either. So failing a phase 2 trial, if on all parameters, obviously will cut you any sort of funding for later on, so you can't even dream of going to a phase 3. But having some kind of success in phase 2, people would expect you to move to the next stage. People wouldn't unless you have an unlimited amount of money, and then you can repeat another phase 2 to understand another mechanism, etc. Now, it also occurs that in many of our phase 2 trials, we have explored many different angles to the question, and hence, what is a phase 2 and what is a phase 3 actually? I think it's also a question we need to ask ourselves. We are currently running two phase 3 trials, one in Europe, one in the US. Big pharma may consider those as phase 2 because it's 240 patients. Is it a phase 3? Is it a phase 2? I don't know. The only thing I can say is that if it does meet the predefined endpoints, then we have a good chance of getting closer to the market.
Donna, you in phase 2, phase 3? I forget where you are.
Phase 2 and phase 3.
Phase 2
a
and
number of
phase 3.
programs in phase 3, and this comment about what is phase 2, phase 3 is very important at this time where there are a number of countries where accelerated approval pathways are available so that you can move more quickly into commercialization, and much of that is related to the disease itself. So certainly for rare diseases or very serious and life-threatening conditions, those are areas where there are opportunities to move into commercialization a little bit quicker. I think, I'd like to sort of also add on to the comment about, what we learn from phase 2 trials is critical for further development of programs, and certainly not meeting a predefined primary endpoint in a phase 2 study, to me, would not mean the end of development for that indication at all. And as long as we're able to learn something and we've thought ahead of time of what the product is that we're developing and what our target population is, we need to keep moving towards that target and figure out how the information we have is going to help us make further decisions to get there.
I agree, but it's also important to note that one of the most critical success factors is knowing when to stop and when to quit. So when you have a phase 2 trial that doesn't meet its primary endpoint, if you have a strong scientific indicator that, in a different population with a different design, perhaps with a different important endpoint, you can move forward and be successful, that's terrific. And it's also terrific, by the way, if more neutral observers who aren't as biased as you are agree, because there's a tendency always to think that your phase 2 trial would have been successful, but for one little thing, if you're very caught up in it. But if it's what it's telling you, but if where you come out with that neutral observation is that you may well have a drug that works, but perhaps it doesn't work as well as you hoped, then you have a difficult decision, especially in the area we're dealing in, where a number of these therapies are going to have a significant cost of goods, significant expenses in bringing it to the marketplace. You want to make sure before you invest in phase 3 with that particular asset, if you have a choice, that you have a good likelihood of having not just efficacy, but efficacy sufficient to bring success in the marketplace.
I think that's probably a good point to pick up on. So we've navigated a little bit the phase 2, some science. The startup will probably not quit at that point, but maybe want to pivot and say we learned some things. But then as we think about some products certainly that have gone onto the market in cell therapy, the cost of goods became quite a significant issue, and relative to reimbursement and other things. So at what point should we start thinking about the cost of goods and the mode of delivery? Is that, as we get into that stage 3 and phase 3 trial, as the commercialization team sort of gets engaged, when do you start thinking about that, Gil?
I think you've got to think really early about that. The reality of it is that if you look across the spectrum of clinical indications that the field tends to focus on, it really consists of many of the greatest areas of substantial unmet medical need in medicine today. Things that traditional medical approaches have not developed an effective solution for. That translates to a very high cost of care, a very high quality of life impact on many patients. I think that's where we are seeing and are likely to see our biggest uptake in this next generation of products that are navigating through development successfully and then ultimately are practiced clinically to deal with things like stroke and heart disease and a whole cancer and a whole range of other therapies where traditional medicine just has not really yet had the kind of impact that people have been hoping for a long time. The reimbursement aspect of that, and as it relates directly to things like your cost of goods and your ability to actually deploy in a sensible way commercially and in a scalable way, is absolutely critical. And I encourage people to think about that process very early on. I think most of the people in this room recognize that by the time you enter into a phase 3 study, that you've really got to have your manufacturing approach worked out. Yeah. And not just your manufacturing approach, but you've got to have a good vision for the logistics, the downstream logistics, if you will, of how are you actually going to deliver this technology into the clinical marketplace in a rational, organized, systematic, and reproducible way. And I think that's where we've seen some companies in the past that have been successful in the clinical development front, successful on the regulatory front, and even arguably successful on the reimbursement front, but they didn't connect all the dots in terms of how they were actually going to deliver the therapy in a consistent, rational way to really take care of the reimbursement issues or the mechanics of the reimbursement issues. have run into problems. So we need to be thinking through that process all the way through from start to finish, and we've got to start early in development if we want to avoid some pretty expensive mistakes.
I mean, much of that is carved out really in phase 2 now, because
That's
some of
right.
the phase 2 trials are getting quite large, and you really need to have that target in mind and know that if you're going to meet it with, various approaches.
Yeah.
Christian, I mean, it's a business conference here, so you're thinking about cost of goods and gross margin of the eventual product beyond the science. Is that already in your thinking?
Well, you can ask my team in the audience how much pressure do I put on all of them to control this element. It just adds another risk in your development process. As what we do, our product is the process. So if you're going to change something in the process later on in your development, you run the risk of having to repeat many of the earlier steps you've done before. So and mastering cost of goods is also related to the to your process. So it's immensely important. The risk is because if you look at clinical trials, what we usually see is a certain percentage of patients that are responders, certain percent of patients that are non-responders. And then the outcome of the trial is the percentage of patients that are responders versus the percentage of patients that are non-responders. This translates into a cost-effectiveness equation. This translates into if I have 100% of my patients that respond, I'm going to be able to claim more than if I have 10% of patients that respond. So it just adds another risk element in the development process, which in the past was much less relevant for pharmaceutical companies.
Gwen, that's on the mind of Adaptimmune when you think of the IV bag at the patient bedside of T cells.
Yeah, no, I agree completely with both Gil and Christian's comments, and we've started in phase 1 to think about the commercial process, both from a view of cost of goods and also feasibility of commercialization, because again, the process is the product, and if we change the manufacturing process, we might be changing characteristics in the T cell product that we can't yet detect in our release tests, because we haven't fully figured out how to characterize the cells and how that relates to efficacy in the clinic because we're too early on. So we decided to just remove that variable from the equation and lock down a commercial process early on, repeat a pilot study using that new process to confirm that the cells are still indeed behaving the same, and then take away that risk. Our product is a frozen product, so in terms of delivery, it's an IV, so it's somewhat simpler to deliver to the patients.
Jay, I'll maybe be a little controversial here in this audience, but sometimes I hear from pharma, because of some of these complexities, that pharma would prefer in the end. Well, I like the understanding the mechanism of action of some of these cell therapies, but it'd be easier in a way if I can just, once I've understood that, just go back and make a small molecule drug that has effectively blocks it and does it, and I put it into my manufacturing cells, and I'll just wait for everyone to figure out the science going on here. Are you, the pharma, do you think, open to cope with the different biological manufacturing complexities and delivery of being a bit of a device company, and maybe it's easier for J&J, somewhat of a device and therapeutic and all these cogs issues coming down?
Yeah, I think that, of course, pharma companies are going to differ in that. But we heard that same argument about monoclonal antibodies for many years. We'll just develop a small molecule against the same target. But the fact is that, the types of targets you can access, the types of protein surfaces, the types of activities you can do, the types of specificity you can get with an antibody are very different from what you can get with a small molecule. So I would suggest the answer here with regard to cell-based therapies is very similar. It's important to choose your target to be one that there's good reason to believe cannot be addressed with a small molecule. But there are plenty of things that small molecules can't do. I think it would be risky or dangerous to develop a cell therapy that would be easily supplanted with a small molecule because the risks, the costs, the simplicity, the manufacturing, there's just so many aspects that are attractive, but there's also extremely good reason to believe that there are many types of functions that cells will do best, and if you're focusing on those functions, you're going to protect yourself from that risk. And I think certainly in J&J, we're quite prepared to utilize that modality or any modality of therapy that we think will be the best to address the diseases that we're interested in.
Yeah, I think that's a really important point. I mean, we've seen over and over again over the past decade examples where cell-based therapies are clearly working through multimodal mechanisms, and it's going to be difficult to replicate that with a single small molecule or a single simple biological entity, say a peptide or something like that. There may be instances where people are able to do that, but I think that more and more we see that cells are dynamic living entities. They're actually reacting to the way the body is signaling in multiple different ways in the case of a disease or an injury, and cells are able to respond to that in a way that is fundamentally different from many of the classic therapeutic modalities that people have explored. There will be instances for sure where you can replicate certain aspects of that or use a combination of different agents to deliver something that perhaps is more efficient in some respects. But I think it's not an accident, nor should it be a surprise, that many of the different indications that we're focused on collectively as a field are going to require those types of multimodal responses and interventions, if you will, to maximize therapeutic benefit. I think it also leads naturally to the next question, which has to do with the reimbursement aspect of it, and I think there we can learn a lesson from what Gilead has gone through recently, where they have a very expensive therapeutic regimen that the media actually, there's a lot of noise and consternation about the high cost of that, but the reality of it is that it's curative in nature, and it's eliminating or alleviating a lot of downstream costs. Well, that's exactly how I think many of us are thinking about cell therapy, that you can deliver a much more cost-effective outcome, and it's really central to the value proposition that these technologies are going to be able to deliver at the end of the day. And yes, it may have a very high reimbursement point, but it's going to be worth it in many instances where you can demonstrate that you're not only fundamentally improving clinical outcomes and enhancing patient quality of life, but you're also reducing the overall cost of care in many different areas. But it's incumbent upon us in the field to be looking at those types of things and demonstrating it with data that we're gathering through our clinical studies and our pharmacoeconomic studies and the other things we're doing along the way.
Do you have to start with what that value of the reimbursement is, or do you start with this is a very expensive drug to make and deliver, and I need to therefore justify this cost to be able to make a profit?
No, I think you've got to analyze it very carefully, and luckily there are some outside groups that have deep expertise, some that we've worked with at Athersys, looking at the standard of care in its entirety. For an example, for an indication like stroke, where you've got the time and intensive care, the hospitalization, the downstream physical therapy and rehab costs, the impact on quality of life, and there's a whole chain of events that occur that in its totality add up to an enormous burden in terms of the clinical burden, the quality of life burden, and the cost of care burden. So being able to factor that into the thinking so that we can say, hey, here's what we're looking at and here's what the therapy is actually delivering is very, very important. And I think building that into your analytics early on is critically important so that by the time you're actually sitting down with the regulators and the reimbursement agencies or representatives, there's already a lot of common knowledge and a lot of analysis that you can point to and have a common conversation about, hey, here's where the value of this is really going to be. I think it's important to start that early on. A lot of people ignore it and they start doing it very late in the process. I think that's a mistake.
Yeah, I would second that. We have what we call and a lot of companies do, a target product profile process. And from early in development, we specify more as we go through development, but assessing the marketplace that you're going to launch into, not the current marketplace. What other competition is likely to launch? What's the standard of care? What's the standard of reimbursement? What's a likely reimbursement? And what are the value drivers is important early on. It speaks to a number of issues. First, the cost of goods issue. You can get a realistic view of what's an acceptable cost of goods, what's a target cost of goods, and understand whether you actually have a reasonable proposition. And as you, and I certainly agree with the other speakers, as you look to lock into a process early on in your development, particularly if, as several of our speakers mentioned, you have some hope of launching after a phase 2 clinical trial. But also, it helps you design your clinical development plan, because if you understand what are the value drivers, what are the benefits that patients, physicians, and payers value, then you can design your program to address, and your product and your trials to address those benefits and to show whether you have those benefits. And you can also, as I alluded to earlier, with that plan together, make the tough decisions to stop before you've spent hundreds of millions of dollars when it's pretty clear that you're not going to you may have a product that could get regulatory approval, has risks that outweigh benefits, but the overall picture is just not going to be a success in the marketplace. And if you don't understand what the market is likely to look for, you can't make that determination.
Gil, you're operating your business out of Europe. Do you have to look, as you're doing clinical trials in two places, also the regulatory reimbursement out of what Europe looks like and a cost of a insured life for the lifetime compared to what we have here in the US, and think differently, or do you think this will converge as some of the regulatory is converging?
Well, I think there's first a discrepancy between regulators and insurers and reimbursers, because they're not looking for the same things. Yep. And you're going to have to address one and find a way to address the second one, because as you said, if you just get the product approved and you don't have a reimbursement, and there are some on the European markets, and you're not going to be successful commercially. And then there is also a discrepancy between what the U.S. and what the U.S. and what Europe wants, and the endpoints are different. Frequently, when you go and talk to reimbursement agencies, they say, "Well, I don't really care about how much the patient is going to cost me 3 years down the road. If your one-time cost at the beginning is that high, I just cannot afford it." So all that discussion basically highlights the fact that we, as industry, have to take things into our hands and make sure that our cost of products at the beginning is as realistic and as acceptable to society as it can be. Even as a face value, even as a number, as a big number.
Yeah. And I think if I could add that process of developing the target product profile, it's an evolving exercise. So it starts very early on in development, but as you gain information and as there are changes in the field, those all need to be factored in to understand, do I still have the potential to deliver this product, and is it still going to be meaningful? Is it something that's going to be reimbursable? Have there been other developments in the field that change the meaning of this product to the patient population?
Would Mesoblast go for try and get reimbursement first in the US for your product, did you think about it, or outside the US? How do you think what would be first?
Well, we look first at like our key jurisdiction for approvals, and so we'll focus on that area first. But then the other jurisdictions are not really far behind, because most of us are gearing towards global therapies and not regional therapies. So it's all part of the picture, but we do start usually with one, and that's usually the U.S.
Yeah.
But it's fair to say in some cases, other jurisdictions may provide you an opportunity to get there faster.
To get there
right?
faster,
Look at
right.
The recent JCR meso submission in Japan, taking advantage of the new regulatory framework there. Mm-hmm. So it creates some very exciting opportunities, I think, for companies to think a little bit differently about the order of development and where the shortest path to a commercial market may be, and then think about their reimbursement strategy accordingly in terms of the staging of various things and how they can take advantage of the exciting new regulatory initiatives, whether it be in Japan or in Europe or increasingly in the U.S. as well.
Gwen, do you rely on GSK and your panel to worry about reimbursement? Is that,
Well, we haven't gotten that far yet, but we have very early on again, and this is linked up with the cost of goods, thought about how expensive will it be to create the product and what is likely reimbursement that we could do. So more than a back of the envelope calculation, we certainly did go out to the marketplace and see, what is the target patient population and how expensive is it to treat them, and what is the likely reimbursement, and then how much does our product cost to make. So it's feasible. And then we look at how we can reduce the cost of goods and the manufacturing costs, and we put together a long-term plan. So there's near-term ways to reduce it in long-term, and that's as far as we've gotten so far. But some of these later, things that you've all mentioned, we're about to enter into. Yeah.
So we have about 10 minutes or so left on the panel. If any questions in the room that people feel to address. I don't know if we can get a microphone up the back there, or if you can speak loudly, we can hear your question, I think.
I'll speak loudly. How's that? So the panel, I feel that there's been a tremendous, you haven't discussed a very important area, and that is intellectual property. So I'd like you all to comment perhaps on intellectual property, not only patents, not only patents and trade secrets, but also branding of your particular product. Would you please comment on intellectual property?
Well, it's kind of a broad question, I guess. I mean, there's a lot of different ways you could think about the IP question. I think what we've seen is just in kind of a general sense over the past few years is that there's been good, steady, consistent growth in terms of IP in many different forms relating to composition of matter, specific formulations, methods of use, manufacturing-related technologies, and a whole bunch of other areas. And I think that the field and the rules of the game, if you will, on the IP front are continuing to evolve, and people are thinking about ways to how do I protect the product, and then that gets to the second issue, not directly related, but I think the second question, or at least the second thing embedded in your question, relates to the branding aspect of it. And again, just as there's going to be lots of different complexities and lots of different ways to think about IP strategies and the like, there's going to be lots of different ways to approach the branding strategy. Some companies are focused more on, we want to deliver an off-the-shelf solution that's right in the hospital pharmacy that can be delivered easily at the point of care, whether in frozen form or whatever it may be, so that the clinician can use it right when they need to, and they don't have to worry about how it's going to be administered to patients. It's literally call up to the pharmacy and say, send me a unit of whatever it is I need, so I can give it to the patient in a predictable, kind of streamlined manner. Others are going to be more process-oriented, and of course there are complexities embedded with that, but that doesn't mean that those technologies are going to be any less valid. I think they're going to be equally valid, and it's really just a question of the indication, and it's working out the logistics of how is it best delivered clinically to the patient and through the doctor in the way that is rational. So, it's hard to comment with specificity on what it is you're actually getting to, other than to say it's a complex landscape and it's continuing to evolve around us, and people I know are thinking about that in terms of how they manage their IP strategies and the like.
Thank you. Is it on? Yeah. So, Brian Lee from PBS Biotech. I think we touched a little bit with Gail and Jay about the manufacturing challenges for this complex product, unlike the small molecule or therapy proteins. But this is much more complex product. There's manufacturing challenges in terms of the security of your supply chain and consistency of quality, and how are you going to address this complex product, and what would be a solution for that?
Well,
much simpler than the IP question.
It's complex, all right.
It is a reality.
It is a reality, and it's also eye-opening to think about how things change from, the hospital lab and, the bedside, prepare for a stem cell transplant, and what it takes to actually get into industry and then commercialize something and have consistency. And so that consistency piece of it is critical. And I think, as everyone knows, this is a growing field of a very complex specialty where there are a number of manufacturing organizations that focus specifically just on this matter. But it is an important strategic imperative to understand what your product is, how you can consistently manufacture it, lock down like your critical supplies as well, and have that all scoped out well in advance of your proposed launch, so that you can consistently deliver the product. I think it'd be nothing short of a disaster to get to registration and not have commercial supply. So, I mean, all these pieces of the puzzle need to fit together appropriately on the timeline, so that we're ready to deliver these therapies that we've determined have a real value to the community.
Yeah. Well, sorry, go ahead, Jeff.
Well, I'd add that in order to ensure a consistent, high-quality product, it's essential to understand what are the critical quality attributes. Even in something as relatively simple as a monoclonal antibody. And even in 2014, much more so 10 or 20 years ago, products change, shift, drift for reasons we can't always explain. when we upscale, when we open a new plant, when we change a supplier of a raw ingredient, when we tweak the process to increase productivity, we wind up with a product. Sometimes we can measure the differences, sometimes they're less apparent. It's pretty predictable in a cell-based therapy that any of those things will change the product. And it's pretty predictable that we won't always know because there is no way we can fully characterize a cell-based product. So critical to their development is understanding what are the attributes of the product that are essential for its efficacy and safety. And that involves a deep understanding of its mechanism of action and the science around what it's doing. And it may involve, in animal models, hopefully, sometimes in humans, making variations of the product to understand which of the things that the cell can do, express, or secrete are critical or are not critical. That's a lot of what some would call basic research, but without that, the ability to upscale and to ensure to your satisfaction, not to mention regulator satisfaction, that you can make that product in large quantity for large numbers of patients over a significant period of time, it becomes a guessing game.
And it's not just the making of the product, it's how do you actually deliver it from where you make it into the supply chain to ensure that there's good product integrity the whole way through. The good news there is that the companies that are focused on developing the products don't necessarily, and I think don't expect, to have to solve for all the steps along the way in that process. We're working with other companies or other groups. I mean, look at Life Technologies and Thermo Fisher in terms of they have for years been developing, validating, and then delivering solutions, practical solutions, technologies that are enabling to the rest of us in the field that say, well, look, I can't take the time to figure out how am I going to develop a shipping container that's fully validated that allows me to make sure that my product is the same as when it left point A and got to point B, and I can rely on that 100% of the time. But thankfully, there are great organizations out there that are developing those solutions and are working with companies to say, okay, let's take a look ahead. Let's take a look at what the next generation technology needs to be so that we can make this as practical and as efficient as possible. There are contract manufacturing organizations and other groups out there, Lonza and others that are great examples of companies that have spent many years trying to develop and refine technology, learning from various things, and then optimizing from there. And in some ways, we're really standing on the shoulders of the early giants, Genzyme. I mean, I've heard, in fact, many people here, I'm sure, have heard many presentations that they made early on talking about the steps they needed to go through to figure out how do we get the product in an early cell therapy form from point A to point B and do that consistently. They had to solve for a lot of things, and now, thankfully, a lot of the rest of us can actually build on that and not take it for granted, but learn from that and apply the wisdom that they generated over time.
Good question.
My name is Jill Helms. I'm from Stanford University, and I wanted to come back to your point. So I do believe that understanding the mechanism of action allows a much cleaner path forward. But the tools and technologies that we've developed at research institutions to address these exact mechanisms of action, I wonder if they're going to be accepted by regulatory bodies. Right, so there are fancy genetic models that allow you to track the cells regardless of where they go in the body. Will the regulatory bodies accept these kinds of models versus the typical studies that you do in a dog, for instance? What do you think about that?
Well, I think so. Where the data are there. I was a regulator for 20 years and headed regulation of biologic therapeutics, including cell and gene therapies, and antibodies and proteins when the science around them was much less important. And I think that regulators, the regulation of biologics is science-based. There's a vast number of scientists. When I was at CBER, it was located at the NIH. It's now moved, but still composed of substantial core of scientists who are interested in new technologies. I think you'll hear over the course of this meeting from regulators who can answer that question better. But I think if the data are there, if the science is there when you're moving into a new area, there's a lot of openness to use of, especially for the types of uses you're talking about. For understanding, when it comes to demonstration of efficacy, you want to see clinical benefits. When it comes to surrogate endpoints, there'll be a lot of debate. But when it comes to understanding mechanisms and what cells do and how to characterize them, yes, I think there's a great deal of openness to that.
Jay, maybe you can comment on ARM's engagement with FDA and other regulatory groups as part of the strategy for actually addressing those types of issues as well, because I think that you've obviously been an integral part of that, and but maybe you can kind of add some color to that.
Well, right, and I think the FDA has shown a lot of openness and reached out to ARM in that regard. So our technology and science groups, as well as our regulatory groups, do interact. And I'm thinking back through a history. There was the use of PCR when it was new, for example. The use of a lot of technologies that were not there. The agency was very open to their use for product regulation in the technology development product. So I think the openness will be there, and I think we've seen that openness, and I think there's a lot of work, to your point, Gil, point of your question, going on now to understand what are the best new technologies and the best ways to deploy them in a general basis. Of course, when you have product-specific technologies, then you need to deal with them in a product-specific basis. Important.
Mark, you started the conversation this morning extolling the virtues of cell and gene therapy and all the advances. And I'm just curious for the panel, it seems to me there's a disconnect that society as a whole has no idea. They think this is the future, 20 years, 30 years. And if you look at, for instance, the program on Sunday night on the high price of cancer therapies that was on 60 Minutes, I've seen more about this in the last 48 hours. This is not news. I mean, they've been expensive for a long time. I'm just wondering, how does either ARM or the individual companies, how do you view that we can turn the discussion towards the successes and towards the here and now and away from all the downside or the lack of progress?
Well, I think that first we need to have a very big success. That's the first question. Once we have a very big success, then all the other questions will be able to be addressed more easily than they are today. And we're getting there. I think the immunotherapy field, the regenerative medicine field, we are getting to a point where we will be able to show to the public in general that our data is solid, robust, and provides significant clinical benefit to patients. The cost question is real. I mean, we cannot evade it. It is real. It is what it is. And our role is to try to manage that as well as we can. But I don't believe in the public forums that I'm currently seeing that there is skepticism. There is debate because people confuse embryonic stem cells, iPS, genes, cells, etc. And it's a complex thing to talk about. But once you focus on one specific therapy, if it's very successful from a clinical perspective, and I think most of those questions will be answered relatively easily. That's my belief, at least.
Yeah, but my take is this is exactly why ARM has an entire team of people that are committed to identifying areas where we can communicate more effectively, and that means, and why we have patient advocacy groups that are involved in ARM, because it's important for them to not only be aware of what's going on, but also to have a voice and be part of the process. We need to do a better job of educating the media and educating the outside world about the successes. It's sad, but it's true that the media tends to focus on writing stories about the things that are most controversial, most inflammatory, because the old philosophy is that that's what's going to draw people to buy the newspaper. And I think we need to do a better job of actually pointing to not only the future successes, but the successes that are already out there. And there's plenty of those. And I think that sometimes we don't do a good enough job marketing what we've already accomplished, as well as the things that we're poised to accomplish in the years ahead. And I think we need to do that. And trust me, the public has shown a consistent hunger for wanting to know about new medical advances and new technologies. And we have to have a consistent, persistent mentality that we're prepared to educate the world outside about the really exciting stuff that's going on and why it matters, and how we can not only change medicine, but also deliver solutions that deliver a lot of value to the healthcare system at large. Because like it or not, that's the era that we're living in. We have to take that into consideration, as Christian was saying.
With that, our time unfortunately is up. I want to really thank our panel members for a great, wide-ranging discussion on many of the hurdles I think we all have to face as an industry. But I'm I'm optimistic the progress we made in the last years. The science is making progress. We think about what we learned from the clinical trials. We think ahead to the commercialization, the cost of goods, and then ultimately the reimbursement and the other issues that were raised in the questions here. I think we're on a good track. So thank you, everyone, and look forward to the next part of the session.